All right, welcome back, everyone. We hope you had a chance to stretch and refresh. We will now move on to the Session 1 Q&A panel discussion, where we'll explore these topics further and address your questions. As a reminder, please submit your questions via the MS Team chat. If you do not have access to that chat, please send your email to orsevents@fda.hhs.gov as is displayed on your screen right now. Please make sure that you put the subject line as AGDD 2025 and who that question will be addressed to. We'll be monitoring this and hopefully we can answer all of your questions during the session. In addition to our original speakers, Dr. Kumari, Dr. Kamal, Dr. Han, and Dr. Kelchen, I am pleased to welcome 3 additional panelists. We have Dr. Wenlei Jiang, whom you briefly heard from this morning, Dr. Zhen Xu, who serves as a Senior Staff Fellow in the Division of Bioequivalence 3 within the Office of Bioequivalence Office of Generic Drugs, and Dr. Xiaoming Xu, who serves as the Division Director for the Division of Pharmaceutical Quality Research 5 in the Office of Pharmaceutical Quality Research in the Office of Pharmaceutical Quality. Thank you to all of our panelists for being here with us today, and we will now take our first question.
Thank you, Maria. So we have our first question for Dr. Kelchen. An RLD for a topical product has 2 approved strengths, but the Ph.D. does not include a waiver approach. Is the waiver for an alternative strength an acceptable option, even if it is not in the Ph.D.?
Thank you for the question. Yes, it's possible that a waiver approach may be applicable for that product, even if the waiver approach isn't already in the product-specific guidance. As I mentioned at the beginning of our talk, we develop and revise the PSGs based on interest as well as availability of resources. And so it's possible we just have not yet incorporated that waiver into that particular PSG. So if you're interested in moving forward with a waiver approach for a product that's not in the where the waiver approach is not in the PSG already, we encourage you to come in and talk to us as part of the pre-ANDA product development meeting request so we can discuss the relevant components of the VE waiver approach, as well as the recommended strength and those sorts of details before you initiate your study. Thank you.
Thank you, Dr. Kelchen. We have our next question for Dr. Naheed Kamal. How do we balance Q1 sameness requirements with the biological excipient variability in generics?
Can you hear me? Thank you for the question. So from our understanding from the research that we have performed to address the challenges associated with the product-specific guidance development, our understanding is the Q1 sameness can be redefined by developing a comprehensive comparative characterization method for the collagen materials and also obviously for the drug performances, so that this approach, the systematic approach, the framework will allow us to see that not only even though the collagen has the biological variability, even though having that is not, it doesn't compromise the therapeutic performance of the test product in compared to the RLD. I hope that answers your question.
Thank you, Dr. Kamal. Our next question is for Dr. Rangeeta Kumari. What are the major offices involved in the STT program?
Thank you. That's a good question. So as I mentioned in my presentation, the SME testing process involves 9 offices, sub-offices within CDER. So 2 super office of generic drug. There are 4 sub-offices within Office of Generic Drug, which Office of Bioequivalence, Office of Generic Drug Policy, Office of Research and Standard, and Office of Safety and Clinical Evaluation. Those are the offices with sub-offices within Office of Generic Drug. And within Office of Pharmaceutical Quality, we have the folks from Research, Office of Pharmaceutical Quality Research, from Policy, Office of Product Quality Assessment. All of us together discuss the complexity associated with each complex NDA that goes through that process.
Thank you.
Thank you.
So we have our next question for Dr. Wenlei Jiang. What types of internal research the STT program focuses on?
Yeah, Thank you for this question. So I think based on our presentation today, you can see actually the STT program focused on reverse engineering and product characterization type of research to really understand like the drug substance, to understand complex drug substance, complex excipients, as well as complex product. And actually, Nahid, Dr. Nahid Kamal today present a very interesting case on the collagen implants to characterize the excipients. I would say for the STT program, it didn't really focus on any in vivo studies. For example, like how to design more efficient pharmacokinetic study or clinical endpoint studies, or any kind of modeling and simulation efforts. Yeah, I will also let Xiaoming chime in on this.
Yeah. As Wenlei mentioned that, and also Rangeeta kind of presented, the part of the program is looking at what is a gap in understanding that wouldn't or prevent us or delay the development of a guidance. So the key focus of the research is really to understand where there may be lack of evidence or where there will be challenges in determining the path forward. That's where the type of research project that would focus on. So as of now, as Wenlei mentioned, a lot of the effort has been putting on the characterizations, reverse engineering, understanding the product from a characterization standpoint, how those measurements may help support the development of or demonstration of the bioequivalence. But I think we have been discussing internally as well. Over time, as our research program become more mature, we'll perhaps expanding the research scope to also including those that may support eventually regulatory assessment. But at the time being, the focus of the research program is really on those that are critically needed for us to produce evidence to fill the knowledge gap in order to develop the guidance. So again, you have seen two cases this morning, but they are representing kind of a fraction of what the both internal and external research effort as part of the GDUFA program. For a broader sense of what type of research is conducted, I think the best resources would be the GDUFA Science Research Report that OGD and OPQ publish every year. I think around there you can find more information about all the critical area of research that have been conducted both internally and as well as externally through contracts. Thank you.
Thank you, Dr. Jiang. Thank you, Dr. Xu. So we have another question for you, Dr. Xiaoming Jiang. If FDA published publications, do you expect us to use that method for our drug development?
Yeah, that's a great question. We oftentimes hear from by interacting with the externally that there seems to be this perceived conception or notion that anything published by the FDA is mandatory to be used, which is not the case. I think particularly for research, I think what's important, we recognize that we, both within FDA and externally, we strive to generating understandings by conducting research. And so the finding that we produced represent our best effort of the current understanding. So we share that knowledge through publication, through a venue like this workshop, but that's only, again, one side of or part of the information that we think would be sufficient or necessary to support a product development and approval. So I guess in short, the publication that published by the FDA researchers are only for, again, share with the outside what is the thought process behind, what are some of the evidence that is used to support some of the guidance recommendations, but by no means those are the rigid recommendation or specific methodology that you have to follow. I think it's more important to view those as the information that would help you to understand some of the recommendation behind rather than rigidly following what is put forward in the publication. Again, those publications are peer-reviewed, so we also encourage also generic industry to conduct your own research, and you can come forward with questions or recommend or an approach that may differ from those recommended in the guidance or those differ from the work that's published by the FDA. Again, the proper venue would probably be the pre-ENDA meeting approach. So yeah, we welcome you to bring those approaches forward, and hopefully you also find the publication useful for your development as well. Wenlei, do you want to add anything?
Yeah, I think we always encourage innovation. FDA has done our part. We also encourage generic applicants to do your part and come up with new methods and share with FDA. Yeah, I think that's about it. Thank you.
Thank you, Dr. Jiang, and thank you, Dr. Xu. We have a follow-up question for you, Dr. Xiaoming. How is FDA communicating the research finding for the internal work? And how do we find out if there is a specific publication for our specific PSG?
I think one-stop shop probably would be the annual GDUFA research report. That would be a summary of all GDUFA research that supported by user fees, and then that may have supported the development of guidances as well as supporting the regulatory assessment. I would say that the GDUFA research report would be the most comprehensive place to stay up to date. But that's published annually. So I guess if you are more interested in to stay alert on the more kind of recent development, I think follow some of the researchers from the FDA probably would be a good idea. And again, there's a lot of the information findings are shared through peer-reviewed articles. So those will appear in the typical scientific journals that it's hard to link them directly to one specific guidance. But you would find a lot of the methodologies described perhaps could be suitable for not just one, but multiple type of products. So hopefully that answered the question. Thank you.
Thank you, Dr.
Wenlei,
Xu.
want to add something?
Yeah, I also direct everyone to the Center for Complex Generic Drug Research, the website. They also have a quite comprehensive collection of the workshops and also the publications from OGD and OPQR FDA. I think the SBIA website is also a good place to look for the past workshop collections. Those are all venues for efficient communication of generic drug research. Yeah, thank you.
Thank you, Dr. Jiang. We have a question for Dr. Zhen Xu. Does the agency allow for biowaiver option when the nasal or orally inhaled products include multiple strengths?
Thank you for the question. So right now, we actually don't have the PKB study, in vivo PKB study waiver option for the orally inhaled and nasal drug product. This is because first the PK dose response, dose proportionality often not established for the multiple strengths of the nasal and inhalation product. Second, the in vitro performance, aerosol performance is often not well understood if they can be scaled to multiple strengths. And also multiple strengths of the IOD are sometimes not proportionally, formulation proportionally made. In that case, currently we don't have this option. But if the generic company, if you believe your drug product qualify for the waiver option, you can submit your proposal and rationale, and we can discuss, for example, the pre-ENA meeting or control correspondence. Thank you for the question.
Thank you, Dr. Zhen. We have another question for Dr. Han or Dr. Zhen. What is the minimum number of inhalations recommended by the FDA?
So the minimum number usually we refer to is that based on the sensitivity of the analytical methods. For example, like the Ph.D. study, you use the minimal number of inhalations that allow you to detect the minimal deposition and of the drug product. Also for the dissolution study, the minimal that the inhalation numbers sometimes refers to you do not use too much too many inhalations that cause the agglomeration that deposited in the different stages of cascade impact. That will impact the sensitivity of the dissolution study.
Thank you, Dr. Han. We have a question for Dr. Megan Kelchen. If an applicant has an approved generic for a high strength of a topical product, can they receive a waiver of a BE study for the low strength?
Thank you for the question. If you have an approved drug product already for one strength and you want to use the waiver approach for the alternative strength, I think that's something we would entertain, and I encourage you to, again, come in and talk to us in a pre-ENDA meeting. As I mentioned, for example, for the IVRT method validation studies, if there's significant time between developing the higher strength and the lower strength, there may be a need to repeat some of those method validation studies. So I think it would be beneficial to come discuss with us about what kind of data we would need and what kind of studies might need to be rerun. But that's definitely something that we are amenable to and would be willing to discuss about. Thank you.
Thank you, Dr. Kelchen. We have a question for Dr. Naheed Kamal. How can in vitro methods replace an in vivo bioequivalence studies for complex implants?
Question? Yeah, if the in vivo study is difficult to perform, in that case, maybe agency need to rely on the in vitro B demonstration for the drug product. And our research already successfully showed that it is feasible to the in vitro characterization, feasible to address in vitro characterization based, the challenges associated with the drug product when it's all the parameter critical quality attributes, including the in vitro drug release collectively is used. Again, if that's filling the in vitro BE approach, filling that gap or not, regarding that, it depends on there's a review concern, because the package needs to submit adequate comparative comprehensive characterization data between the generic product and the RLD. And as Wenlei and Xiaoming mentioned, we always encourage the agency to have innovative methods to enhance our understanding, or to strengthen the framework of the in vitro characterization approach. I hope that answers your question. You guys want to answer something? Xiaoming.
So it's fairly challenging to, I think, determine if in vitro should replace in vivo assessment, because the challenge arises from the differences in the complexity of the type of dosage form, route of administration, and also the sensitivity of what we're trying to measure in terms of determining the bioequivalence. In cases where in vivo data, the sensitivity or variability is highly variable, insensitive to what is critical for the an equivalence measure such as in Nahid's example, that the drug is intended to work locally after the surgery, then the systemic measure of the PK variability can be high, or in another case, if it's inhalation product, again, locally acting that measures systemic PK could be challenging. That's where I think cases can be made that in vitro measurement can provide higher sensitivity. But then the challenge become, is that change or sensitivity really appropriate for the determination of the equivalent? So, which is a secondary, I think, challenges or concerns. But overall, I think we recognize the fact that some of the more advanced in vitro method measurement currently can provide better sensitivity with lower variability, which may provide us more information in terms of determining as a totality of evidence approach what would be considered equivalent comparing a generic product to the reference product. So I would add to what he's mentioned is I think it's definitely case by case of the product. For one product may be suitable, for another product maybe not, and even for the product that may be suitable, also depending on the type of evidence that's provided as part of the submission. So it really depends. But in general, we do recognize this is a viable approach to support the generic development for the areas, particularly that is challenging to develop. So, thank you.
Thank you, Dr. Xu, and thank you, Dr. Kamal. So we have a question for Dr. Han. Where a generic RLD has an indication for multiple indications, example, asthma and COPD, is it sufficient to select 2 inhalation profiles for the RA-PSD test, or would more be required? Example, 2 for each indication. What approach should be taken to justify the selection?
So that's a good question. Honestly, we haven't met this case before. And I think that is so usually, for example, like the product has 2 indications. In the past, for the clinical endpoint study, so for example, has 2 indications for COPD and asthma. Usually, we just recommend that the clinical endpoint study be conducted in the patients with asthma, because asthma, we think is that the asthma patients may be more sensitive to the clinical efficacy demonstration. So in your question for the real risk APSD, the 2 indications. So if you need 2 inhalation profiles from each indication, and now we cannot give you a definite answer, but that we recommend you that can submit a meeting request or a control correspondence that we will provide more answer to that. Thank you.
Thank you, Dr. Han. We have a question for Dr. Zhen Xu. For suspension nasal sprays, such as fluticasone nasal sprays, can we do dissolution study using bulk suspension instead of emitted dose?
Thank you for the question. So the dissolution study, the purpose of doing dissolution study is the API of the dissolution becomes rate limiting in the drug absorption. So from a bioequivalence perspective, I think the dissolution study should be conducted using the spray instead of the bulk solution suspension, because the drugs, the suspension nasal spray, such as the fluticasone propionate, it's defined as a complex drug product. Not only because it's a combination product, device and the formulation combination, but also it's the suspension itself is heterogeneous nature. In that case, the dissolution study using the spray instead of the bulk Makes sense. Also, if you look at all other recommendations or other in vitro studies, like single iteration content, droplet size distribution by laser diffraction, and spirit pattern, plume geometry, and also the small particle size. All these studies, they use aerosol spray instead of the bulk solution, bulk suspension. Thank you for the question.
Thank you, Dr. Zhen. We have a question. Our next question is for Dr. Wenlei Jiang, Dr. Xiaoming Xu, and Dr. Rangeeta Kumari. How do we measure the impact of the STT program and demonstrate that the published Ph.D. reduce industry inquiries and improve quality of the submitted ANDAs?
Yeah, I can go ahead to start. I think that's a very good question. Actually, we are collecting data to develop matrix to measure the impact of STT program on the complex product PSG development. I can think of, for example, we can measure the time between the approval of NDA and the date of the publication of PSG. I think that can be a good metric to measure how STT program impacts efficient complex PSG development. But I have to say, this may not be a kind of very straightforward causal effect relationship, but it will be a good indication of how STT program kind of helps efficient PSG development. Yeah, I will stop here, and maybe we can go around to.
Yeah, I think ultimately is to the applicant, to the industry, is it helpful for you? Ultimately, can we get your product coming in through first cycle review to getting approved? And how effective is the recommendation providing the PSG that help you to accomplish that? So I think it's that's our long-term goal. How to measure it, as Wenlei mentioned, I think we're trying to develop some internal metrics to help us understand. But we do see, at least in the short term of about 4 years now running this program, we do see increased collaboration across offices within CDER, and improved communication internally, which in the long run should help with both sharing the understanding externally through the guidances, as well as ultimately help knowledge transfer between research policy and the review, which ultimately benefiting you as industry. So hopefully this is something that, again, we see as success, but we're still not yet done with the GDUFA 3. So looking forward to GDUFA 4 to see how we can become more efficient.
Thank you, Dr. Jiang, and thank you, Dr. Xu. We have a question for Dr. Megan Kelchen. What kind of BE studies can be waived as part of the waiver approach for the topical products?
Thank you for the question. So for the characterization-based approach, the bioequivalent studies that we see in the as part of that approach include an IVRT study, and then in some cases an IVPT study or a PK study. So as you saw in my presentation, an IVRT study is an important component of the waiver approach. And so we typically cannot waive the IVRT study as part of the waiver approach. So where the waiver approach may be applicable is for products that have 2 strengths but include a recommendation for an IVPT study or in some cases a PK study as part of the characterization-based approach. For the in vivo options, we typically see a waiver for the comparative clinical endpoint study, but a waiver of a VC study may be something that we could also explore. Thank you.
Thank you, Dr. Kelchen. We have a question for Dr. Han. Thank you for your nice presentation. You mentioned that the use of PBPK as supportive studies for OIDPS. Do you consider PBPK-based VBE a surrogate for in vivo BE? If so, what are the requirements for the model?
Good question. So before you think that you can use the PK modeling as a surrogate for the in vivos and the PKB study, you probably need to provide the following information. First is your justification. Why you think that you can use the modeling that instead of the in vivo PKB study. And that's your justification. Second, I can think is that you need to provide enough information on your modeling. For example, how is your modeling, if your modeling is adequate, and especially importantly, if your modeling has been validated. So after you have all this information, I suggest you to submit a meeting request and provide with all the detailed information, and we can discuss it give you more feedback on your modeling during the meeting. Thank you.
Thank you, Dr. Han. Now we have a question for Dr. Zhen Xu. The BE evaluation of APSD data is based on the PBE statistics. Does FDA have better ways to compare APSD data between T and R?
Thank you for the question. We noticed that there are different approaches, statistical approach for APSD studies. Like FDA, we use PBE, population BE for the impact size mass statistics. We also do MMAD and GSD, mass median error diameter and the geometric standard deviation, and also fine particle mass, PBE analysis as a supportive evidence. We noticed that some other regulatory body like EMA, they use average BE, 90% confidence interval between, within the plus minus 15% acceptance limit. So, yeah, so different regulatory body have different recommendations. So if you consider your application using a different statistical method, you can discuss with us, FDA, and propose your ideas in statistical analysis in either pre-ENDA or control correspondence. Thank you for the question.
Thank you, Dr. Zhen. We have another question for you, Dr. Zhen and Dr. Han. Are there circumstances where the optional computational model would be requested during the review to supplement the BE evidence, where an applicant has followed the approach stated in the PSG and met any acceptance criteria included?
As I remember, I can remember that one case is that in one application, the applicant submitted the PKB studies data that without a chock block. And during the review, we think that we need to know that the PK profile from the inhaled portion of the drug in the lung. And we said talk with the applicant then submitted in the modeling that to show that they can use the partial AUC instead of doing a whole new set PKB study with chock block. And their models that they discuss with our model to show that the model is adequate, and the partial AUC from the PKB study without a chock block can be used to represent the data from the chock block PKB study. And after discussion, and we think it may be reasonable for them to do that. That's the one case I can remember.
Yeah, so I think if we currently have all these new PSG's recommendations. If there's anything that's deviate from those recommendations, we recommend you to talk with us and submit your pre-ENDA, or we like to hear your thoughts and your proposed ideas. Thank you.
Thank you, Dr. Han, and thank you, Dr. Zhen. We have our next question for Dr. Wenlei Jiang. EMA applies different approach for the BE evaluation of the APSD data. Any thoughts on potential alignment?
Yeah, that's a very good question. Maybe that's a question for Dr. Xu, but I can go ahead to start. Yeah, as Dr. Xu just mentioned, FDA and EMA apply different approach for the evaluation of APSD. Actually, in general, FDA and EMA apply different bioequivalence approach for the evaluation of orally inhaled drug products. I think FDA, we have this totality of evidence approach, and the EMA, they have this stepwise approach. And down to APSD, and one is population bioequivalence, the other is average bioequivalence. I think it's very important to have some discussion between EMA and FDA on these approaches. Actually, in the past GBHI conference, like this topic has been discussed. I think first, we really need to understand each agency's approach, like some of the specific steps, like which moiety is measured and what are the step-by-step process to kind of compare the APSD. So I think this is a good question. We can initiate some discussion between the two agencies to really understand each agency's current approach and see any potential for future alignment. Yeah, I will leave it to you.
I just want to add a little bit details about the APSD statistics. So I think one of the key issues for APSD analysis is the we call the profile analysis. That means to know the entire distribution instead of just the one spot. In that case, we can compare test the reference in the entire picture instead of just one spot. So like the FDA's recommendation using the mass median error diameter and the geometric standard deviation, we assume it's log normally distributed. So in that case, the entire distribution profile can be reduced to MMAD and GSD. So these two parameters can represent the entire profile. So when we do a PV analysis of these two parameters, we can define the entire profile. So it's similar to what the like EMA proposed. Their 90% confidence interval. If it's log normally distributed, the acceptance limit is between 85% to 118%. So I think we can still discuss details, compare with different regulatory bodies, the APSD analysis, and see if we can have common sense. Also, in FDA, we evaluate in our BE review, we evaluate the profile of APSD using the modified chi-square ratio analysis. Sometimes the answer, the evaluation outcome agree with the PV analysis of ISM. Sometimes not. Sometimes they need to have justifications. So yeah, it's the APSD study, the APSD data. It's a lot of information we like to discuss with you and get more valuable feedback. Thank you for the question.
Thank you, Dr. Jiang, and thank you, Dr. Xu. We have a question for Dr. Han. Could you provide comment on whether any additional data may be required to supplement that included in the PSG, and when assumptions on the Q1, Q2, and device as listed in the PSG are met? For example, are there circumstances when CFD data may be requested?
So CFD is modeling is tools that simulate the local deposition in the lung. And I can give you one example of use CFD data to support the BE. Let's hypothesize that you have marginally failed the real-risk APSD data. Yes, that says some difference between the test and the IOD product of the APSD data. But you can present your CFD modeling data to show that this difference between the test and the IOD products in the realistic APSD actually does not matter. That also does not affect the local deposition. So, and in combination with other data, for example, the PKB data and the others that the morphology or other in vitro tests. And overall, we think that your CF data may support the BE establishment, even though you've marginally failed the APSD study. That's the one example I can give. Thank you.
Thank you, Dr. Han. Now we have our last question for session 1 Q&A panel discussion for Dr. Megan Kelchen. Can you speak more on the findings from the TDS research that was presented?
Thank you for the question and the opportunity to expand on that point. So as part of that internal research, again, we have reviewed the 30 ANDA for TDS products that came in during GDUFA 1 and GDUFA 2. And we looked at the inactive ingredients that were used in those products. And we also looked at whether those irritation or sensitization studies failed. And what we ultimately found was that there were a few failures for the irritation study and only one failure for the sensitization study. And when we took a deeper dive into those applications, we saw that the failures for the irritation study were primarily due to study design rather than specific aspects of the test product. So it wasn't necessarily related to an inactive ingredient, but rather an underpowering of the study or something like that. And then for the sensitization study, we found that the inactive or the active ingredient for that drug product was actually known to be a sensitizing agent for the skin. And so we actually ended up revising that PSG to remove the recommendation for a sensitization study. So I think in summary, our conclusions were that based on this limited data set that we had, the inactive ingredients did not necessarily play a role in the irritation or sensitization study failures. Thank you.
Thank you so much to all of our session 1 presenter and panelists for sharing their expertise and insights. This now concludes session 1 and our first morning of this 2-day workshop. Let's now take a lunch break, and we will reconvene at 1 p.m. Eastern for session 2, Novel Bioequivalent Study Design and Recommendations. We remind you to please continue being a part of our discussion and submit your questions. They can be submitted via the MS Teams Q&A option, and if you do not have that option, please email orsevents@fda.hhs.gov. We will be continuously monitoring this inbox and hoping to address your questions during the designated Q&A panel. Thank you so much again to all of our speakers and presenters, and we hope you enjoy lunch, and we'll come back at 1 p.m.